everyone. I hope everybody's doing well. I'm Justine. I'm a first-year master's student working at the Douglas Research Center, and I'll be moderating the talk today. So it's my great pleasure to welcome Kevin Whittingstall for today's CIC lecture. Dr. Whittingstall is a full professor in the Department of Radiology at the Université Sherbrooke in Quebec. He received his master's and PhD in physics at Dalhousie University in Halifax and completed postdoctoral studies at the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. And his main research interests are in the development of non-invasive MRI tools for measuring and interpreting brain function and structure in humans. And in particular, his lab focuses on the balance between neural activity and cerebral blood supply, so neurovascular coupling, and how disruptions in this balance are related to diseases of the brain. And from 2011 to 2021, he held a Canada Research Chair in neurovascular coupling. So thank you very much, Kevin, for being here today. Just a quick reminder to everyone to keep the mics closed during the presentation. And as we said, so I'll be monitoring the chat. If ever you have questions, you can write them in there and we'll go through them at the end of the presentation. And Dr. Whittingstall has also agreed to hold a student discussion at the end of the talk for any more open discussion questions. So anyone who wants to can stick around for that as well. So with that, Kevin, thanks again for taking the time to give this lecture today, and the floor is yours.
Wonderful. Thank you very much for that nice introduction, and thank you for the invitation. And to all of you who took time out of your busy day to come hear me talk about things that are important to me in the lab here at the Université de Sherbrooke. So once this responds, okay, so we'll just get right into it. Blood supply to the brain is extremely important. And proof of that is just by looking at the way the brain evolved to irrigate this organ. So basically, we have three major arteries coming off the heart: the left and right internal carotid arteries and the basilar arteries. These three arteries then bifurcate into literally millions of tiny blood vessels, which end up perfusing these very hungry neurons. They can consist of the large surface vessels. These are the ones that really ride along the cortex. And then from time to time, they'll penetrate at 90 degrees into the cortex to go feed these hungry neurons. And so when we put all this together, blood vessels and neurons, we typically refer to it as a neurovascular unit. So it's the communication between blood vessels and neurons. So typically when we think about blood flow and blood vessels, the first thing that comes to mind are different pathologies related to the absence of blood flow. And the first one being, of course, stroke. Stroke being the number one or number two killer on the planet. But the lack of blood flow also has other effects. If it's not a complete lack of blood flow, you can generate hypoxia in the brain, or the lack of oxygen. Now, the brain can't stockpile oxygen. So as soon as there's a reduction in O2 levels, you may not have a full stroke, but different symptoms may start to appear. And we've known for a long time that these vascular components are quite critical in different neurodegenerative disorders. Even a fairly large paper coming out of the MNI that found that vascular features may actually be some of the earliest biomarkers in age-related cognitive decline or dementia or Alzheimer's. So today we're going to look at a relatively simple question that we think we'd have an answer to, but it turns out that it's actually extremely complex. And that question is, what controls blood flow to the brain? More specifically, how on earth does the heart schlep blood from three arteries to a specific brain area without schlepping it somewhere else? So typically when we think of blood flow and blood control, two things come to mind. The first is the concept of autoregulation. which basically means that the brain likes to be in a constant state. It doesn't like deviation. So basically, the blood flow will tend to be constant in the brain in the face of changes in blood pressure. And the reason this happens is because the diameter of the blood vessels in the brain, more specifically the luminal diameter, will adapt in the face of changes in pressure in order to maintain a constant blood flow. Once you exit this realm, then you can have either a collapsing of a blood vessel or hemorrhaging. Now, the rules governing autoregulation are still a bit unclear, but again, anatomy can inform us. Here's a stain of the basilar artery, and the red arrows here, these are actually really high caliber intracranial nerves that ensheath within the endothelia of the large blood vessels. So here's a schematic on the right here. You see that the large blood vessels on the cortex seem to be controlled directly by these large nerves. You can think of when you breathe a lot of carbon dioxide, the reason these large blood vessels dilate is because of these, in this case, the trigeminal nerve, most likely just to flush out the excess carbon dioxide, given its toxicity in the brain. But once we get to the level of the neurons, the rules change. Now, these tiny blood vessels are not so much innervated by the cranial nerves, but by the nerve cells themselves. And so here, in particular, in blue, you have the glial cells, the astrocytes, whose end feet will wrap around these blood vessels, and via communication with the neurons, will trigger vasoactive substances to constrict or dilate the blood vessel in absence of changes of blood pressure. So you can have a large change in arterial diameter despite no change in systemic blood pressure. And so this is the concept of neurovascular coupling. Here's a nice example here. We see an active neuron, and then a few seconds later, after its release of vasoactive substances, particularly in vessels exhibiting smooth muscle, you'll see a light vasodilation, which ensures that the blood can now reach the neuron. Remember, these neurons consume an enormous amount of oxygen during a local field potential or action potentials. And so, keeping them well fed is very, very important. And so this whole concept of vasodilation being related to neural activity is basically the cornerstone of the functional neuroimaging revolution. So 25 years ago, we found out that by putting someone in a very conventional MRI scanner, we have them do some cognitive task, and we found that blood flow, blood oxygenation, or even deoxyhemoglobin levels can change and provide a map of functional activation. And so this has given rise to fMRI, functional MRI, which is now basically the de facto measure that we measure brain activation in humans. It's non-invasive, it's relatively easy to use, and the maps are just absolutely splendid. Now, the issue, of course, is that for every study that shows a really nice relationship between neural activity and blood flow, there's also an equal counterpart. There are papers showing that, in fact, the two measures may not be related to the same level that we thought they were. Some papers show you can have large changes in blood flow without changes in neural activity, large changes in neural activity without changes in blood flow. And so most of these studies are done in animal models, but in humans, this was relatively unknown. And that's because the methodologies that we use in animals, of course, cannot be used in humans. In humans, we're left with non-invasive methodologies. And so I think to set up the next couple of slides, we'll show why neurovascular uncoupling or why understanding it could be important. If we take the case of autism, one thing we've seen over the past 15 years is that when we record blood flow responses in autistic adolescents, the response to stimulation, say visual stimulus, is not that different than non-autistic adolescents. So fMRI or blood flow is not a great tool to differentiate these two cohorts. But if you repeat the same experiment using a very old technology, the electroencephalogram with electrodes affixated to the skull, and show the same stimulus, well, then you see a massive difference between autistics and control. In fact, in the autistic adolescents, you see a reduction of roughly 50% of the EEG signal. And so why is that? Why is it that in EEG, you see a massive difference between the two, but in fMRI, you don't? And what's causing this difference? Which one is right? Which one is wrong? So to answer this, the first thing we did is developed ways of recording EEG and fMRI at the same time. So the first thing we did during my postdoc training is we conducted some of the first simultaneous surface EEG and intracranial recordings in alert primates doing various behavioral tasks. And what we found is that the surface EEG signal, despite being far away from the neurons and relatively lo-fi in terms of technology, was a highly accurate marker of neural activity. So EEG does do a really good job of measuring these extracellular signals. The nice thing about EEG is that you can couple it with MRI to do simultaneous EEG and MRI measures in humans. And so we did some tests, we developed some denoising algorithms and found that we could reconstruct the EEG signal in the MRI environment to essentially the same degree that we would get if we were recording outside the scanner. And so methodologically, it's now possible to record the two simultaneously and have really accurate data. So one of the first things we did is look at the response of the EEG signal to just a typical visual stimulus. Normally, when you present humans with visual stimulation, you'll see an increase in high frequency activity or the gamma band in the few hundred milliseconds following stimulus onset, and a decrease in the low frequency rhythms or the alpha band. So we simply took these rhythms and then correlated them to the bold signal that was recorded simultaneously. And what we found is that for the gamma band, the high frequency signals, that correlated really well with CBF in the primary visual cortex, so the early part of the visual hierarchy. But the alpha signals, they correlated best with CBF signals in the extrastriate areas, in the later visual areas. So right off the bat, it was clear that neurovascular coupling might change depending on where you are in the brain. In one area, it might correlate with one rhythm. In a different brain area, it might correlate with another. The next thing we decided to do is say, well, let's focus on one brain area. And so here we showed healthy human subjects, just a very simple visual stimulus where we increased the contrast and reproduced a well-established finding that the gamma rhythm increases with higher visual contrast. And that can be seen here in the occipital maps. And the bold signal did exactly the same thing. So brighter stimuli, brighter bold, higher EEG, there's nice neurovascular coupling right here. But then what we did is we spatially randomized the stimuli. That is, we just randomly started swapping the position of the pixels while leaving the overall contrast the same. So the brain was still receiving the same amount of information, but it was just deconstructed now. So when you decorrelate the input falling on the retina, what happens is you start decorrelating the neural signals. So they don't sum to give a very large signal anymore, and so you see a decrease in the EEG signal. But for the bold, on the other hand, what we found is that the bold just kept responding with the same amplitude. So by simply changing a very basic characteristic of the visual stimulus, we could turn neurovascular coupling on or off in one brain area of a healthy human. And in fact, by using some computational models, we found out that the EEG signal was basically highly dependent on the synchrony of sensory inputs, whereas the CBF and bold signal, he didn't care so much about whether or not the signals were synchronized, but just how many inputs were falling on the retina. And so this is going to cause a huge dissociation between the metabolic demand, which just cares about how much input it has to deal with, versus the EEG, which cares how well timed these inputs are. So we just kept, beating at this horse and seeing how far we could take it. So we developed one more experiment where we decided to try and trick subjects and play with their levels of anticipation. So here what we did is subjects would fixate on a black dot. That black dot would become red. And 2 seconds after the dot became red, we would show them a high contrast stimuli or a low contrast stimuli. But in some cases, no stimuli would show up at all. So there was no visual input. And so, of course, when there's no visual input, we measure no EEG signal. But when we looked in the visual cortex in terms of bold, we nevertheless saw an increase in the bold signal, despite the fact that there was no visual input falling on the retina. And so when we look at the pupil using measures of pupil diameter, we found that, of course, when you present the retina with visual stimulation, the retina will contract. But when no stimuli was presented, we saw the pupil actually dilate. And we think this is because they're anticipating the arrival of a stimuli that never arrives, which probably activates the cholinergic system, which might have a direct pathway to the vasculature, but not so much to the neurons, at least not enough that we can measure it with EEG. Last but not least is the concept of retrograde vasodilation. So this time, instead of using EEG, we took images of the cerebral blood vessels. So this is not bold or CBF, it's literally in white here. You have the 2 posterior cerebral arteries feeding the visual cortex. And this is what the arteries look like when the subject is resting quietly. These are the same arteries in the same subject, where now the subject is watching a movie. So we're stimulating the retina. And you see here the yellow arrow, the blood vessel is visible during the stimulation, but it's not visible during the baseline condition. And that's expected because we expect there to be vasodilation and an increase in blood flow. What was unexpected, though, is that the vasodilation was not only limited to the visual cortex, but we actually saw it almost 70 millimeters anterior to the site of visual stimulation, very, very, very close to the midbrain, which might indicate that that vasodilation that you see during sensory stimulation is never local. It actually conducts itself up towards the more distal sites. So here's a schematic of what might be happening here. You have 2 pools of neurons, in red the active neurons and in blue the inactive neurons. The neurons activate, vasodilate locally, and then there's retrograde vasodilation all the way up to the major feeding artery. The inactive arteries, of course, will not release these vasoactive signals. However, in the place where there's strong neural activity, there's going to be an increase in blood flow and an increase in metabolism. So that's going to give a bold response. But in the area that was not activated, there's no metabolism, but it's still going to have an increase in blood flow because of this retrograde vasodilation. So it raises this interesting theory that in some cases it might be possible that inactivated areas of the brain might have a stronger bold response than those that are activated due to the lack of metabolism, but an increase in blood flow. And so we're currently conducting some experiments to show that this might in fact be happening even within the visual cortex. So if we bring it back to our initial model of why can there be a dissociation between hemodynamic and neural signals, well, I've just described 4 of them. You have the anatomical variations, depending on where you are in the brain, the rules may change, synchrony versus input, neuromodulation, and retrograde vasodilation. And so after 20 years of studying neurovascular coupling and 10 years as a professor, it started to bug me that maybe the thing that I was best trained in turned out may have not been true. Maybe measures of blood flow are not indicative of neural activity. And so then the question is, well, what do you do? If you no longer believe in what you've been trained your entire life, do you just give up, or do you keep going on, right? And so I decided, well, there's still a lot of science to do, so let's try to turn this into something positive. And so one thing that at least we felt that wasn't really addressed in the question of what controls blood flow was a far more basic concept, which is, what do the blood vessels themselves look like? Instead of focusing on the neural activity, let's look at these blood vessels. Are they even capable of transporting blood to a brain area? And so we decided to look at whether or not dysfunction of the blood vessels themselves could explain different types of neurological disorders. Now, we've known for some time that vascular features of the brain show up before pathology, but we were curious to go a bit further and find out, well, what aspect of the blood vessel could be changing? Could it be something like atherosclerosis that we see in Alzheimer's disease, where there's a buildup of fat inside the blood vessel, which stems the flow of blood? Or could it be something in young people like multiple sclerosis, where chronic inflammation is so high that the brain, the blood vessels start creating new blood vessels because it thinks that something is going horribly wrong. And so you have rampant angiogenesis in the brain. Both of these two things, by the way, could in a strange way cause reductions in blood flow. So the first thing we did is went on Google and said, well, let's find a way to segment blood vessels, and really couldn't find anything. So we started slowly but surely developing some software that would take whether it's a brain image tailored more towards the venous side or the arterial side, and try to segment these blood vessels. The first attempt at doing this was published a few years back, and the code is available online if you want to play with it. And one of the first applications that we took these methods and we looked at the venous vasculature. So we focus only on the position of the cerebral veins. And what we found anyways was that the bold signals that we measure were heavily biased by the position of these veins. So whether we were looking at two brain areas in one subject or the same brain area in two subjects, once you correct for the fact that the size of the blood vessel and its proximity to your site of activation, we found that a lot of our differences disappeared by simply correcting for the vasculature. And it didn't matter if we were looking at task-based fMRI or resting state fMRI. We found that the amplitude of the bold signal could be in fact predicted by simply looking at the SWI image, the venous image of the subject. And then we found that even the timing of the bold response was affected by its proximity to a large vein. So once timing is being affected, well, that means the connectivity between two brain areas is going to be affected. So it didn't matter how you collected your bold data, whether it was task or resting state, correcting for the vasculature, the venous vasculature, seemed to be quite expensive, quite important, excuse me. The next thing is we looked at the map of these blood vessels. So here on the right is the venous density. So we just literally count the number of blood vessels we see and divide by the unit area of the gray matter mask. And what we found is that the layout of these blood vessels was not homogeneous. We found that the vascular density was actually exceptionally high in some of the most popular brain areas, otherwise known as the default mode network. So the bold signal that you measure in these default mode areas is going to be just more sensitive to the vasculature than other brain areas. Now, whether or not that's a cause or effect, that's for later studies to find out. So then we slowly start creeping into the arterial side. So we said, well, okay, blood vessels affect bold. Let's look into different pathologies. So this is work from my PhD student, Charles-Antoine Barbeau-Meunier, who mapped arterial vessels in healthy controls and young women with multiple sclerosis being a state of chronic inflammation. And what we found is that we could see more blood vessels in the multiple sclerosis image than in the healthy controls. And so when we control for things like gray matter atrophy and head size, all these things that could explain these things, we nevertheless saw that the vascular density was always higher in multiple sclerosis compared to healthy controls. And that was pretty much the same no matter where you looked in gray matter, but in white matter, the effect was still significant, but to a lesser extent. And so whether or not this is because properties of the blood are changing and it's causing us to see more blood vessels, that's always a possibility. The other possibility is that what we're seeing here is a rampant angiogenesis. The blood vessels are just proliferating or have been proliferating for a long, long time, which is why we see more branches coming off these arteries. Now, all this is good, but the problem is that as soon as collaborators sent us some images, we always ran into the same problem in segmenting these vessels. And it's a problem that we didn't really know what to do with, and it was the concept of thresholding. At what point do you decide a blood vessel is a blood vessel and not just background noise? And MR angiography images are unique because the signal that you measure is going to highly depend on the angle of which the slices are acquired. This is not the case really for T1 and T2 star images. So the odds of every technologist always acquiring the brain with the same angle is virtually zero, which meant that if we stuck with threshold-based methods, we would not be able to do large studies as you can do with T1 weighted images. So that was the first problem. The second problem is that even if we got rid of that threshold problem, you would end up with a binary map of blood vessels. And we didn't want that. We wanted labels. We wanted the middle cerebral artery to be a different number than, say, the posterior cerebral artery. And we wanted that because at the end of the day, if you want to analyze 1,000 images, you want to transform every image into an Excel spreadsheet. You want to be able to extract metrics from each individual artery, the same way you would with, say, tools like FreeSurfer or Civet, which will take the brain and parcellate it into different areas. And then you can go and extract diameter measures from these different brain areas. So how do we do this? Well, for the better part of 1 year, we got together with some vascular neurologists, neuroradiologists, neurosurgeons, and neurologists. And the students in the lab, we started coloring. We started manually annotating the intracranial arteries on various MRA images from public data sets, from images taken here. And we quickly realized that it's actually quite easy to annotate these arteries because the circle of Willis, the major entry point of these arteries, there are rules. Certain arteries are allowed to connect with others, but other arteries are not allowed. So it's something a human can do really easily, but it just takes a long time. And this is exactly what artificial intelligence is really good at, is replicating what a human can do, but just do it really much faster. So we took all these annotated images, roughly about 300 of them, and we fed them into a basic neural net. And we tried to learn these connective rules such that you could take a TOF MRA image, put it into the neural net, and it could just automatically segment the cerebral vasculature. And that's exactly what it does. Here's an example of the coding that we use right now. The software will spit out 18 different intracranial arteries per image. Here's what the raw image looks like, and here's what it looks like after it's been segmented. Qualitative assessment, we went online and took MRA images from 2 data sets that were not included in the training phase of the algorithm. And overall, we found that there was roughly between 90 and 95% accuracy for the large arteries. For the smaller arteries, the so-called communicating arteries, the accuracy was at roughly 80%. But this was really dependent on field strength. We found that provided you had a 3 Tesla scanner, the accuracy seemed to be well over 90% for most of the arteries. In terms of quantitative assessment, we then compared the prediction that the software spits out to the actual annotation by the expert and computed an overlap or a dice score. And we found the dice scores ranged between, say, 0.5 and 0.8, which initially we thought were quite low. But then when we asked 2 radiologists to annotate the same image and compute the dice correlation between those annotations, turns out that our software is doing exactly what humans can do. And in other words, our segmentation of the arteries is just as good as human experts. The thing that we were most proud of, I think, is that for the large and medium arteries, there was no dependence on image signal to noise ratio. So it didn't matter if you had a 1.5 Tesla or a 3 Tesla image or what orientation you had acquired it, the software will always give you the correct answer. For the communicating arteries, however, there was a slight benefit to using 3 Tesla scanners. The last thing we did is a retest analysis. So here, these are 10 subjects, each of them scanned 4 times, 2 scans in the same session, the remaining 2 sessions on a different day. And we had a dice correlation, an interclass correlation of roughly 95. So you see scan 1 versus scan 2, whether you're computing the volume of the artery or the diameter of the artery, you essentially get the same result in the same person. A interclass correlation coefficient of 0.95. Just to put that into context, the modern day free surfer results has an interclass correlation of roughly 90%. So in terms of test retest, we're in the same order of magnitude as popular gray matter segmentation tools. So now we have the circle of Willis. Here's our raw angiography. Here's the circle of Willis, the large intracranial arteries. And then all we do is we segment the entire vascular tree by simply flood filling into the remaining vasculature. Now, the nice thing about this is that a blood vessel now is only going to be labeled a blood vessel if it can connect back to the circle of Willis. So by keeping that rule in place, you remove all of these false positives that we had before using traditional thresholding. So now there's no threshold needed, and it can segment the entire vasculature while maintaining its label, while maintaining its anatomical label. And so all this is the work of this handsome go-getter right here, Félix Dumais, who worked tirelessly for 2 years, doing everything from the annotation to the deep learning protocol. It's a software that we call Express Intracranial Artery Breakdown, or eicab. The code will be available very shortly online. Basically, it takes any MRA image, no matter its quality or orientation, and within 5 to 10 minutes, depending on the type of computer, it will spit out a labeled image of all arteries, from which then you can choose to look at the intensity as a function of its distance, the diameter as a function of its distance, and so on and so forth. One addition that we're adding to the software is the work of Maxime Chagnon, MSc student in my lab, who's now taking it one step further. So now we have the arteries segmented. And if you look here in the yellow arrows in the anterior circulation, you see there's a faint discoloration at 2 points. This is due to luminal narrowing in someone that was diagnosed with cluster headaches. Thunderclap headaches. And so this luminal narrowing here is a problem. You can see it nicely when you plot the diameter as a function of the distance here. You see these transitions right here. And so what Maxime is working on is trying to automatically isolate these sites of luminal stenosis. And we think stenosis is a big problem because it's going to increase resistance and reduce flow. And so this has been known for a long time in terms of cognitive decline. We know that luminal stenosis is a very, very strong predictor of cognitive decline later in life. But very, very recently, even in the field of psychiatry, we're now starting to rethink whether or not luminal stenosis may have a role in different psychiatric disorders. Again, we're not talking about complete absence of blood flow, but just light hypoxia induced by subclinical luminal narrowing. So then we decided to go straight into applications. This is work from my PhD student, Samantha Cote, who decided that she was going to investigate the role of sex hormones on the size of these blood vessels. And so at the beginning of her PhD, she did this absolutely phenomenal work where she recruited otherwise healthy, normally cycling women and scanned them at 3 times throughout the menstrual cycle in order to isolate times in the follicular phase, so when hormones are low, and the luteal phase when hormones are high, and look at how the brain responds to these 2 states. And the first finding that that surprised us, and despite all the suggestions from the reviewers when it was submitted to try and find an effect, we found that estrogen and blood flow had a very, very low correlation. And if there was a correlation, it was limited to the occipital lobe, but it looked like it was driven by one of these outliers. On the other hand, what was unexpected, though, is that progesterone, which is normally only present in the luteal phase, had a very strong correlation with blood flow to the brain. It was an inverse relationship. So women who have a lot of progesterone seem to have lower blood flow. But this is where mapping arteries becomes really important, because when we looked at where the correlation was highest, here in the blue areas, without knowing the layout of the arteries, we would have said something like anterior cingulate, medial prefrontal cortex. But in fact, this follows the trajectory exactly of the anterior cerebral artery, that major artery that loops up to feed the frontal lobe. So is it possible that progesterone may have an artery-specific effect? Maybe progesterone prefers the anterior artery more than, say, the posterior artery. And if that's true, then this might be opening the possibility of certain pharmacological treatment that can target a certain brain area, but not the other brain area. Because she works tirelessly, she decided that, well, that's a really nice study for endogenous fluctuations in hormones. Well, what about exogenous fluctuations in hormones? And so this is a study that she is carrying out right now, looking at adolescents undergoing gender-affirming hormone therapy. So throughout the therapy, we scan them before, during, and after. And once again, what we're seeing here is a large increase in serum testosterone that is administered over a 6-month period, causes a slight reduction in blood flow, and it looks like the diameter of most blood vessels. So again, this concept that elevated hormones may put the brain in a low CBF, possibly vasoconstrictive environment. The technology also spearheaded some studies that were not possible before. These are two separate studies that were both funded by the National Institutes of Health in collaboration with Columbia University, where here we're looking at the effects of prenatal cannabis and antidepressants throughout the course of pregnancy. So here, for example, is an image of a fetal brain here at roughly 36 weeks. But here below, I think it's still the case, what you're seeing is the world's first image of the intracranial arteries in a 36-week-old fetus. And so you can see that the circle Willis is almost fully developed in this 3D reconstruction right here. So we invite mom in, we scan mom's brain, and we get baby's brain throughout a pregnancy. And then, of course, we follow them later on in life. And so the cool thing about collecting these sorts of data is that you have fetal, neonatal, and adult images. You can now start creating some trajectories of how these arteries develop in life. And the first thing that is surprising us, this is an N of 6 so far, is that the internal carotid artery, the ICA, which is the blue line here, seems to develop much faster than the other intracranial arteries. Of course, when we look later in life, we're looking at different people. But in terms of fetus and neonatal images, this is the same participant that we would be following for 5 years. The other interesting data point from this study is that out of every 10 pregnancies, between 1 and 2 will experience different types of hypertensive disorders. This will be hypertension that a woman experiences throughout gestation. Now, typically during a normal pregnancy, the blood vessels will slightly vasoconstrict. We think it's because of a reduction, or excuse me, an increase in stroke volume. But now if you add hypertension on top of that, it might reduce the caliper of the blood vessels such that the shear stress, computed here in newtons, might be quite high in these women. So this might put the woman at risk of either stroke or preeclampsia or the worst case scenario, eclampsia. And so this will be interesting to look at how the status of or how being pregnant might affect the blood vessels, not only during that 9-month period, but what happens in the postpartum period. Okay, so I have a few, so I'll just finish with the last study that we've been working on. This is a work from Vivian Lee and Félix Janel right here. When Felix joined the lab, he told us that his favorite artery was the anterior corodial artery. So it's this tiny branch coming off the internal carotid artery. And it's quite unique because it feeds, it supplies the anterior portion of the hippocampus, while the posterior portion of the structure is fed by the posterior circulation. So in one way, the hippocampus is lucky because it's well protected against ischemia because you have both vessels coming in, but the anterior part does not have any collateral flow. So what we did is we went into the Oasis cohort where we had 411 MRA images, and we did a follow-up a few years later and looked at how this artery was changing. And at baseline, the first thing we found is that the left ACHA was positively correlated to the mini mental state exam, which is a typical evaluation that is done in cognitive decline. So bigger artery, better cognitive score. But the same was not true for the right ACHA. And when we corrected for different cardiovascular factors, that result still held. So then at follow-up, we decided that we would only analyze the cognition of these patients. And so what we found is that out of 411, 141 were cognitively stable 3 years later, whereas 20 of them were in significant cognitive decline. And so we went back and looked at their baseline MRA images and found that the baseline volume of the left ACHA could predict cognitive decline. In fact, those who had a small ACHA were 4 to 5 times more likely to develop cognitive decline. When we did the same analysis with the hippocampus, on the other hand, we found that it did not have that kind of predictive power. Now, to be fair, here it's the whole volume of the hippocampus, whereas the left ACHA only perfuses the anterior portion. But again, it gives this insight that the size of a blood vessel is most likely a biomarker for something that is going wrong. So why is the ACHA small? One reason could be because its parent artery, the internal carotid artery, is small. So here's a subject who has a reduced luminal diameter of the left, but not the right. The other explanation could be cerebral small vessel disease. So these would be hyperintensities that are seen on FLAIR images. Maybe it's possible that that disease has kind of like taken hold of the left hemisphere relative to the right hemisphere. So when we looked at these images, we found that the volume of the ACHA was entirely determined by the diameter of its parent left ICA. So perfusion of the hippocampus, even though it's far away, might actually depend on how clean the left ICA is. And when we compared it to small vessel disease or hippocampal volumes, we didn't find very good correlations. So it kind of brings us back to where we were 100 years ago, when vascular surgeons started kind of alerting the community that, hey, you know, when we look at the carotid arteries, there's a real asymmetry here. The left carotid seems to be having more of an effect on overall cognition than the right. And so we're quite happy to see a lot of recent papers coming out now where we're starting to analyze brain images, but always keeping the heart nearby. So these are studies that look at patients with heart failure and how heart failure can selectively damage the hippocampus. And just by chance, because the hippocampus is being fed by the ICA. And so now we're in collaboration with Harvard Medical School, where they acquire some really nice MRA images that don't stop at the circle of Willis, but go all the way down almost to the aortic arch, which will allow us to do a full analysis of the heart and brain. Okay, so if there's 3 points from this talk to take home, the first one will be that we think that hemodynamic signals are not accurate markers of neural activity, and that's okay. I mean, we should just call it for what it is. It's a hemodynamic signal. It doesn't necessarily have to be tied to neuronal dysfunction. And if you want to properly interpret this hemodynamic signal, you have to take the structure of the nearby blood vessel into account. And the last thing is some shameless promotion of this software that will soon be available online. It works just like FreeSurfer. You put an MRA image in, and you get colorful images out. And so I'd like to thank you for your time, and thank all the students for making life in the lab just absolutely spectacular. And of course, the funding bodies. And I thank you very much.
Great, thank you so much. That was a super impressive talk, very interesting and very clear. So if anybody has any questions, you can go ahead and write them in the chat or just raise your hand. I can start off with one of my own. Maybe it'll leave time for people to come up with some questions. I was just wondering, based off all of this great work, what are your next steps? Like, what are you planning on working on?
So we think, I mean, we talked about many things, pregnancy and different neurological disorders. What we're now looking at is the concept of a micro-infarct. So these are very, very, very small strokes that usually you only see when combining 3 different types of images, or at least 2 different types of images. We're wondering, so obviously infarcts are bad. We're wondering if a simple MRA image that takes about 3 minutes to acquire, whether or not that image can tell you something about the risk of a microinfarct later in life. And so this is where the lumen diameter of the blood vessel will play a really big role in assessing the health of an individual.
Okay, great, thanks. Cindy, you have a question? Go ahead.
Hi, thank you very much for your talk. I've been searching for images that would show how the blood travels throughout the blood, because when I started to learn that we have these arteries that come up and then dive into the brain, I was thinking this structure of the blood vessels would be impacting the measurements that we can derive from ASL images, like the arterial transit time. And I haven't been able to find like a movie showing this, like just for me to clearly think about what is going on there. And I was wondering what would be the impact, for example, on this flow, on this trajectory of the blood in things, for example, on the fMRI, or even the cerebral blood flow calculation that has been done so far, that not many studies had the possibility to acquire different post-labeling delays.
Post-labeling delays, yeah. I think that's an excellent question. I believe my PhD student, Sam, is here somewhere. And I think she'd be more than happy to talk to you hours on end about this, because this is something that troubles her a great deal. And in fact, what she is going to show very shortly is exactly what you just said, is that if you take an artery and you do a very simple calculation, you compute its distance along that artery, what you find is that that distance is very similar to a map of arterial transit time. In other words, the length of an artery is going to dictate your CBF signal or your ASL signal deep in the parenchyma. And so we think that with a map of these blood vessels, it would be really good to use the distance as a factor in the calculation or in the quantification of CPF. Now, for your question about a movie of blood traveling through the brain, of course, that would be ideal. You can do 4D flow through the circle of Willis. We've done that here. You can reconstruct the tensor and watch blood kind of pulsate through the large arteries. In the small arteries, there's not a lot of signal though. But that's okay. We think that simply reconstructing the trajectory of the artery and getting its distance will be very important in differentiating high versus low ASL signals.
We have a question from Pascal in the chat, wondering if you could just comment on applying these tools to various conditions, for example, with chronic pain.
Yes, that's what the software was developed for. It was to look at pathologies that we think are being dictated by the neurons, but in fact might be due to different vascular remodeling. And so chronic pain, again, with the increased inflammation, could be an ideal model for looking at how these blood vessels remodel in that pathology.
Okay. Rosanna, go ahead.
Yeah, hi, thanks so much for an excellent talk. I'm really interested in this topic. One question I had for you is, I know that some of your early work was very much trying to understand the relationship between, bold activity, EPI related activity and vasculature. And I'm just curious, now that you've discovered so many fascinating changes in vasculature that can occur with different conditions, if you've looked at all into healthy aging, for example, because a lot of fMRI work has, compared a group of younger adults to older adults and shown different areas of the brain. Sometimes you see hypoactivation in older adults, sometimes you see hyperactivation in older adults. And I was just sort of curious if any of the research that you've done or that you know about has sort of spoken to those different patterns of findings. Sometimes you also see more activity, like you see anterior to posterior shifts or posterior to anterior shifts, for example, and patterns of activity with aging. So I was just curious if you you could even speculate if you don't have the data.
I can speculate and non-speculate. So I'm very happy. We're in collaboration now with Columbia University, where we're analyzing, it's roughly 5,000 images from the Northern Manhattan study. It's a population-based study where we have people between the ages of 40 and 95 years old. And yeah, unexpectedly, blood vessels tend to dilate with aging. And it's important to note that that is most likely an adaptive response to atherosclerosis. We think atherosclerosis starts far earlier in life than we previously believed. So we think that the blood vessels will try to compensate by dilating in order to keep the same luminal diameter to pass by. So what happens is that the blood vessels in normal aging will get larger. We know that stroke volume goes down, so the blood velocity is going to decrease. So what happens is that your blood volume seems to increase in the proximal brain areas, but distally, your vessel appears shorter. So it really seems to depend on what brain area you're going to be examining. If you're examining a cortical area that is fed off the early branches of the anterior and posterior, you're going to see different effects than if you're deep in the parenchyma at the tail end of, say, the ACA it goes really, really long. It goes all the way up to back to the parietal lobe. That's where you can see some major differences. So my speculation would be that I think that a lot of the bold signals that we're seeing, their variations across healthy aging, have little to do with neural activity, unfortunately. Or unfortunately, I think it's great news because I think it's a vascular signal. I mean, we should be really interpreting this, but I think a lot
still
of it,
fascinating.
I still think it has to do with just the size and the density of these blood vessels. Yeah.
Yeah, Cindy, go ahead.
I was wondering, for these models that you are creating in your lab to help visualize the vasculature, these models cannot yet fully resolve the micro vessels. What is the implication for that? And I was thinking, for example, with different structural MRI that we are able to derive volumes, we know that this volume is constructed for not only the cells, the number of cells or the volume of the cells or the axons or whatever, but now we should also start to incorporate the fact that we have vessels in there, blood vessels, and as we age, we don't know how is that changing. Since we are not able to visualize them, maybe you are more familiarized with what is known in histology, for example, or what would be the implications if we are not able to visualize them right now?
Wow, okay, well, I think it's, I don't think we're anywhere near seeing small vessels based on intravascular imaging. Now we have FLAIR images, we have SWI images. I think there's hope that we'll see an echo of small vessel disease. But for angiography, which is an intravascular signal, by the time you get to the small blood vessels, blood is just trickling along. The velocity is so low that I highly doubt that even at 7 Tesla, we'll start seeing these really small vessels. I mean, we can see things like the lenticulostriate arteries. So we can see a few perforating arteries, but the small ones, I think ultimately we're going to have to rely on extravascular signals in order to see these. But when it comes to cortical thickness, yes, I think that from here on in, when you compute a cortical thickness map, you need to take into account the position of the blood vessels, because we will shortly publish a paper that shows that when you compute cortical thickness in subjects where you also have the position of the blood vessels, the cortical thickness values are inflated in areas where you have really large blood vessels. And so when you strip these blood vessels out of the analysis, you start seeing that the cortical thickness map starts to change. It's not as different depending on where you are in the brain as it was before. And we think that's because of a partial volume effect. Because remember, in T1 images, if you have high volume, and there's some really nice work from Claudine Gautier's group showing this, it's going to affect the intensity on your T1-based image. So your algorithm might think the cortex is thick, but in fact, that top layer is just the extravascular signal coming from a really large surface artery.
Thank you.
All right, great. So I think we're pretty much out of time. So thanks again very much, Kevin, for this great talk. So this concludes today's lecture. And as I mentioned earlier, anyone who has any further questions can stay for a little bit of a discussion.